It's my great pleasure today to introduce our first speaker for today, Dr. Tala Awada from the University of Nebraska-Lincoln. Tala is an Associate Dean Director at the Agricultural Research Division and a professor at the School of Natural Resources. She co-leads the Nebraska Long-Term Ecosystem Research Network and oversees the establishment of plant phenotyping in Nebraska. Tala's research is in the areas of plant physiology, crop abiotic, forest and grassland ecology, and vegetation cover change as impacted by climate change. Tala received her PhD in plant sciences from the University of Saskatchewan. Today, she's going to talk about the implication of plant phenotyping in plant science research. Please welcome Dr. Tala Awada.
If you haven't noticed by now, she's my young sister. Well, it's good to be back. I haven't been here for many years. I want to thank Morris. I want to thank the organizing committee for inviting us from Nebraska to come and talk about our recent investments in this area and highlight some of the ongoing research that's happening at our institution. I am plant ecophysiologist by training, but most of my work is in the forest and grassland ecology. But if you're in Nebraska, you really can't escape the crop sciences. So in recent years, I've been more and more embedded in the crop sciences, but this presentation really reflects my role, administrative role, in overseeing the facilities and making sure our faculty are collaborating, make an environment for them to collaborate better and to make significant contributions in research. So just a little bit of a background on our facilities. In 2014, UNL made a serious investment, or a decision to make a serious investment in the area of high-throughput plant phenotyping. And this really complements existing expertise on campus related to image analysis and image acquisition in various scales. And the idea is if we can invest in a higher resolution, higher frequency images, we will be able to better extract the information from the optical characteristics of these leaves and relate those to the functional traits and thus the genetic expression. And in this, address some of the major concerns in agriculture, current and emerging issues related to climate change, to anthropogenic management, and so on. So a little bit of a background about our previous, and for many years, these investments have been going on for over 2 decades under the Center for Advanced Land Management and Information Technology. So although they're remote sensing faculty, and although their main aim was not to relate genotype by environment interactions, they made serious contributions in the area of chlorophyll quantification using remote sensing techniques, whether it's proximal or using remote satellite imaging. And they've successfully managed to relate these into net primary production and into NDVI and so on. Many of the instruments you actually use on a daily basis uses some of these algorithms and models that were developed by these groups, like the Anatoly Gitelson and the Ranquist and the Gammons and so on. So for many years, so for many years, over 2 decades now, they've had what they call the backpack program, where they would go in the field with a person with a hyperspectral imaging and collect information, which, as you see, it is, what is that thing? Something is wrong with it. Well, back. Well, we don't need it. So, they were collecting serious imaging that used for extracting and developing of these models, but weren't enough to actually link the genotype to the phenotype. And they've taken advantage. The middle one, which is the tractor, the light isn't working, so I'm just going to describe it. The tractor technology, which was named Hercules, has run for over 14 years over the Ameriflux network system, which I don't know if you're familiar with it. It's a carbon sequestration network. And we have one of the oldest ag networks in Nebraska. And with this, they were able to relate optical characteristics of plants, net primary production, and so on. And they had the aircraft program, which was equipped with a hyperspectral, and now this program is being revamped to incorporate fluorescence in addition to hyperspectral cameras, again, to study vegetation and productivity. Thank you. Thank you. And productivity. So the recent investments we have really comes to complement these expertise and it'll allow us to better relate the genotype to the phenotype. So the first investment, which has been running for around 2 years, 2.5 years, is the HTS LimnaTex canalizer, which many of you are familiar with. It specializes in small plant systems. I will talk in more details in a bit about each of those. The second one was the greenhouse, again, LimnaTech. But the third one, we went a very different route. We went with a spider cam, and it's the second in the world after ETH in Zurich. So, all right. So the first HTS system, which specializes in small plants, has been very useful to study Arabidopsis, whether the epigenetic of it, study mutants, the responses to stresses. And the system is quick. It takes over 100 plants, and it permits to study or phenotype any plant that's less than 10 centimeters. And really, this has complemented the efforts and our collaborations with Lycor Scientific, whereby they do have Arabidopsis small chamber for gas exchange and allows us to bring the two measures together to have a better information on the behavior or responses of plants to stresses. We have 4 cameras in this system, the visible, RGB, the fluorescence, infrared, and near-infrared. And really, we felt that this is the best optimum combination of the two. You can't really go with fluorescence without an infrared. Infrared is a thermal, and really you can detect stomatal conductance much earlier in the process than you would detect fluorescence. So we needed all these complementary images to be able to have a better information on responses to stresses, let's say drought. And our computer, we have a really great team of computer scientists who have been able to analyze these images and validate the new developed algorithms and model against LimnaTech models. Many of us are not very keen with LimnaTech, hopefully no one here from LimnaTech, because of the black boxes that they impose on us. So we really, our scientists, especially computer scientists and engineers, if they can't open this black box and they don't know what it is in it and they can't manipulate it, they don't have much faith in the output of these boxes. So for us now, for graduate students, we feel comfortable of them using the existing LimnaTech algorithms for that specific chamber, but we can't say the same for the other chambers. So the next investment has just been installed in March 2017, 2 months ago. That's why the field is still empty. And we modeled this exactly after the ETH in Zurich. We visited them, and we decided to go with this system rather than a LimnaTech field system because of the flexibility. The flexibility and the impact of the LimnaTech on the light environment above the vegetation. But flexibility was our #1 reason. With this system, SpiderCam is responsible to put the dolly, manipulate the dolly, and give us all the programs, and it's our responsibility to install the cameras and to integrate these cameras within the SpiderCam system, which was great for our engineers and scientists. And given the change in technology on a weekly basis almost, this gives us the flexibility to change cameras, to improve on the system, and always modify it to fit the user's needs in the field. And so with this, the system is pretty simple. SpiderCam, if you don't know, is a broadcasting company. And when I first asked him, how do we know it's going to work? He said, it is going to work because there is zero tolerance for error in our business. If we are broadcasting live and the system goes down, the company is in trouble. So that was what sold us into the system. We had to install, in this case, we have 33-meter tall poles on 4 corners with a winch housing in the middle. And the system can scan vegetation down to 2 centimeters and up to 5.5 meters. Our main target was 6 meters for sweet sorghum, but we had to compromise. It's very windy in Nebraska, so we had to compromise on the height of the vegetation to improve scanning capabilities and quality of images. So in addition, on the field, we have a below-ground sub-irrigation system, drip irrigation system, that can be manipulated on a 35-meter square plots. And in this case, you can independently manipulate soil moisture in each of these, I think around 130 plots in the 1-acre footprint. We do not have any results yet because it's just been installed, but I can show you some of the results from the Swiss collaborators. They have been incredibly helpful. They've shared with us all their blueprints, and they want us to succeed and develop more research with them. So it was really good decision related to this. So they have really published several of these manuscripts already. And in this study, they've just shown us these are several genotypes of soybean, and they have now accurately managed to measure their heights throughout the growing season and separate them. This is false color image, and separate the genotypes based on height. And they have done it, they've related it into reference height and into destructive measures, and they have pretty good relationship. They use LiDAR for this, but they have been now working more into the processes governing the functioning of some of these genotypes, in addition to the morphology and the architecture. So they have a much bigger site. We only have 1 acre, they have 1 hectare. But it's the exact same system. We share some of the cameras, but they keep changing their cameras, and that's part of the flexibility of it. And here is our last investment, which is really the LimnaTech 3D high throughput phenotyping. It's a 30,000 square feet greenhouse. It's been functioning over a year and a half now, collecting quality data. The system allows for 672 plants, a maximum weight of the pots around 10 liters, and we invested in 4 different chambers: the visible, the near-infrared, the hyperspectral, and the fluorescence. So, and the system also permits each, as you would expect, each of these pots has a barcode, and it gets scanned before it enters the chamber, so the computer or the system knows exactly what needs to happen, whether it goes through the 4 chambers, whether it needs water, needs nutrients, and so on, and everything is administered automatically. It takes us 2 days to scan the around 700 pots in the greenhouse if they go through all the chambers. Otherwise, if faculty have other needs or other requirements, we can always accommodate them. So this is some of the outputs that some of our faculty who've done research shared with me. On this, in this case, Dr. Yufang Ji, who is a biosystem engineer, was trying to validate the system. The first year and a half, we really concentrated our efforts on validation and testing the LimnaTech black boxes and developing our own algorithms. I'm not saying we've done some progress, but there is long ways to go as everybody's in the same boat. So in this system, basically, Dr. G was trying to compare two extreme genotypes of maize. One is a mini genotype, and the other is, I think, gross tall, James. And what he's done, he was trying to see whether the system can actually measure the height through destructive measures and through scanning. And he had pretty good relationships, and he tried also to extract water relations parameters from the near-infrared camera and the hyperspectral camera. And he was, through destructive measures and through the scanning measures, and he was pretty successful in doing this and in identifying the differences between the genotype using the greenhouse facilities. And when he related these to destructive measures, there are lots of destructive measures that takes on initially, and he had a very good relationship over here between measured water content using destructive measures and what we got from the LemnaTech system. Same here is predicted leaf water content with measured. We added a soybean, here is only maize. So we have had some few successes using the greenhouse. Now their efforts is focused more, in addition to the morphology and the architecture of these plants, is to looking more into processes. For instance, in this image, same again with Dr. G, he found that in maize, the leaves and the stems have very different hyperspectral signature. And so you can actually now separate these two accurately from the images that you acquired. And we go beyond that. Now they have found that just before flowering in sorghum, changes in the hyperspectral imaging happens. And so is this consistent through several genotypes? Can we predict flowering by changes in these hyperspectral signatures? So these are things that they are looking at. We are now delving into the process aspect of it. Another thing Dr. G decided to do is to test whether these traits are transferable to the field. And he built this little bicycle-based system, basically, where it's equipped with several imaging. But in addition to this, he's trying to incorporate with it robotics, whereby it's automated and it samples the soil and microenvironmental characteristics as it's moving into the field. So in this case, I think he has had some initial successes. It's, again, there's lots of work involved still to happen. He's managed to separate shade leaves from sun leaves, and what's the value for those relative to growth habits and productivity and responses to stresses. It's all being investigated by his lab. Remember, he's an engineer and works with other faculty in the plant sciences to come up with these questions. Another study I want to highlight very quickly, this study is truly interdisciplinary, and it's been going on for several years, and it's a collaboration between UNL, Australia, and several institutions within the US. And basically, it focuses on trying to understand differences between rice genotypes in their water and salt stress. And they had 380 genotypes planted in several places, but also tested at UNL. And the focus was at the initial developmental stage. And here they are trying, and they have succeeded in linking the genetic expression using the gene space like the QTLs and the genome-wide association with the functional characteristics of these genes, with the gene association networks, and relate all these to the phenotypic expressions and get us somewhere. I'm just going to share a couple of slides that Dr. Walia from UNL shared with me. In this study, which lasted maybe 20 days, they collected over 3 million images for the 380 genotypes. And each plant was imaged daily on two side views and top view, and in several different cameras. Of course, you take much more than 2 views, but optimally, they decided that these are the ones that they're going to be using for further the analysis. And sure enough, 3 days after the application of the salt stress, you could see differences between, let me go back, between genotypes, and they were able to identify 2 extreme genotypes in their responses and use those for further analysis. And when they studied their growth response over time, it's sure enough, the blue genotype over here, the Kihogo, I can't pronounce these things, maintained growth while the other one dropped down, basically. So when they used the fluorescence to compare these 2 genotypes, and the darker is the color, the higher is the difference between these genotypes, they found that their photosynthetic system was impacted as soon as 6 days, or differences appeared as soon as 6 days after application of the salt stress. And all this was extracted from using the fluorescence images. And then they related, now I'm talking totally out of my area, but they used the likelihood ratio test performed to identify important SNPs that would relate to these phenotypes. And they identified 115 of those. And he shared with me an example from the chromosome 1, which, if you think about it, that chromosome actually regulates the growth and the ionic responses during salinity, and makes perfect sense for those working in the plant physiology that this would be part of or one of the reasons why you have different responses. And again, they identified in total 35 genes with a probability of 75% that they shared common causal variant using the GWAS analysis. So they concluded that these image-based phenotyping provides really good dynamic response to rice plants, and they can extract genetic-based responses to salinity stresses. I wanna, before I just got the 5-minute note here, I just wanna go quickly about to highlight some of the computing sciences and some of our quantitative results that came up were shared with us in the last year or so. So how do we deal with our data and data management? It's really a team effort. We have what we call the Holland Computing Center, which is a supercomputer. Their job is to make sure we have enough space, make sure that we can access these data, and these data are stored somewhere deep in the system, that 10, 15 years from now, PIs want their data, we can give it back to them. And also to develop the metadata associated with this, because it's a big question, what kind of metadata you need 20 years from now, somebody can actually use these images effectively. And then we have the Plant Vision Initiative, which are a group of scientists from multidisciplinary that work together into answering some of these questions related of extracting information from these images. And then we have the Quantitative Life Science Initiative led by Jennifer Clark. Her main responsibility is to help faculty find ways to analyze these data and develop experimental design to implement these experiments in a good way. So I just want to go through it very quickly here. We have 2 computer scientists that are helping us with what they call holistic approach and component-based approach to analyzing these images. Holistic-based approach is taking the whole plant as one unit and show us what they can extract from it, information, whether it's architecture, whether it's orientation of leaves, aspects, and then the component-based, we're taking leaf by leaf and studying those to answer some of the important questions. So here, for instance, is an example of is basically understanding the aspects of the plant and how it grows by taking always images from a 0 degree and 90 degree and take a ratio of those, so you can have an indication or an understanding of how these leaves are arranged as the plant is growing. The second approach has been to again, holistic approach, looking at the aspect of it, and basically taking the height of the square in the first image with the minimum diameter in the second image, get the ratio of those to get the aspect. So they've developed these models for many of the grass species, soybean. If you're working on soybean, you know it's much more complex than this. But we are heading somewhere. There's some progress being made over here. And what she does, Dr. Shruti Shudhari, is like many of you working in the computer sciences, they take the image, they subtract the part, and then they get a gray image, a binary image, skeleton image, and then they start allocating and following these images. There's lots of machine learning in the system, and you need to know if a leaf has died, that the system needs to know that the leaf died, and I have to continue counting these leaves. So that's an example of how they have accomplished this, and that's an example of how she tracks these leaf emergence from start to finish. And the beauty about our greenhouse is that it can accommodate plants up to 2.5 meters tall, so you can follow plants almost up to maturity, and in many cases to maturity. So where are we at? Where are the challenges? I am hopeful by the end of the 2 days would have figured out would have gotten some questions or answers on how do we address better the scaling issues. These are some of our major challenges. How do we deal with variation in instrumentation and image acquisition and data management, access, sharing, analysis, experimental design, coordination across institutions. I mean, we're all sometimes reinventing the same wheel. How can we coordinate the effort for the whole discipline to move forward? And graduate education, I don't want to forget that because they're mostly doing the work these days. And before I go, if you have time and you're available, consider submitting an abstract to the AGU meeting in New Orleans in December. It's a great venue to have the phenotyping and the agronomist and the breeders to meet with the remote sensing people, because this is their main meeting, and to develop these collaborations. And finally, we have 2 positions coming out. So if you're interested, if you're in grad school, if you're interested in this discipline, we have an open rank faculty position that's going to be announced in the next week or so on plant phenotyping. And another one is the managing for the field facility. Again, is another position that will be available in the next week or so. Thank you so much for your attention. Hopefully it was helpful to you.